Thanks, Sunil, for the invitation to come and present. I came also last year, and it's great to actually be on standing in front today. So I'm the CEO and founder of Ternary Therapeutics. We're a London-based tech bio company with labs in Stevenage Bioscience Catalyst. We're just over a year old, and I'm going to take you through a little bit around the tech platform we built and some of the early traction that we've made. So over the last 5 to 10 years, we've had an absolute revolution in target identification. Precision medicine undertaken on patient level data gives us unprecedented accuracy in identifying the key drivers in disease. But it's quite interesting, isn't it, that a lot of the drug pipelines haven't actually now bursting at the seams with therapies for these drivers of disease. And I think that this is quite a surprising finding, at least when I was looking into it, that 90% of most important genetic drivers of disease are inactivating mutations. They're the wrong directionality to traditional drug modalities. Traditionally, we look at trying to inhibit and degrade molecules, but obviously we need to reactivate all of these. And unsurprisingly, that's reflected in the fact that only 0.1% of drugs can target them. Interestingly, a drug that sits within that bucket is a drug like effector, which only actually originally was approved for around 3,000 patients that have a specific mutation, cystic fibrosis patients in CFTR. And obviously that drug as a monotherapy had peak yearly sales of $700 million and is now multiple billions a year of part of combinations. So there's a massive opportunity for people that are able to unlock this space and find new medicines that specifically reactivate genetic drivers of disease. So a relatively recent modality of small molecule drugs called molecular glues can, in theory, do this. So if we imagine that typical signaling in cells is carried out through protein-protein interactions, and if a mutation is on the surface of that protein, it can disrupt that interaction and prevent the signaling from being transduced. But if we introduce a molecular glue into this, we can re-establish that interaction and correct the disease biology. Unfortunately, all molecular glues to date have the opposite directionality. They inhibit and degrade. Furthermore, to compound matters, molecular glues are very hard to discover. As I mentioned, they're relatively new. There isn't a lot of technologies out there for the rational design of them. And quite often these are discovered by serendipity, and there's very long time frames and it takes a lot of time and money to identify these drugs. So a massive opportunity here for new technologies to really disrupt this space. So what we've built is an end-to-end tech platform for the rational design of molecular glues. We have 4 separate models that address all of the key blockers in space. So Leopard is our target selection tool. It identifies hotspots on the surface of a protein that can be gluable. Puffin is our protein-protein matching algorithm. We have virtual libraries of thousands of protein structures, which we can very rapidly evaluate against a protein of interest, generating and ranking tens of millions of potential complexes to identify the best protein partners that can interact with one another. Gecko can then take those poses and fine-tune them using steered molecular dynamics to get down to the accuracy of crystallography. And then we use those structures to build our virtual screening pipeline, which is Octopus, which is the part that we all care about, which is how we design and optimize the molecular glue therapy. So I did wonder whether this is going to work. It hasn't worked. That's fine. It's fine. I'm going to just skip it.
Do you click it to make it?
I don't think so, no. I think it's because I sent you a PDF.
Oh, okay.
That would probably do it, right? Oh. I'm going to skip straight on and just give you an example of how we validated our platforms. This is a proof of concept project that we ran in collaboration with O2H with the Kickstarter award. So this is an example in the literature of molecular glue degrader where we know the protein of interest and the E3 ligase. But the project was stopped because the ternary complex structure, that's with 2 proteins with the drug in the middle, is unable to be solved. Main reason is because the E3 ligase is over 180 kilodaltons. It's very flexible. You can't even isolate the full length protein. There's no crystal structure for it. It's unlikely to be crystallizable. So this is an example of the limitations that we see quite a lot. It's a novel E3 ligase for TPD. No one's used it before. And there's no understanding of where the small molecule combines with the protein. So really in the dark here. So we used Leopard. We undertook a gluability assessment and the target were able to identify gluable hotspots on the surface. We then did a computational array with over 400 E3 ligase structures, and we identified the E3 ligase that was in the literature as one of the top ranked proteins for our protein of interest. And then we fine-tuned that and we arrived at this structure that you see on the bottom right-hand side, which was our theoretical structure. I should also say that because we didn't have the monomeric structure for the E3 ligase, we had to generate that using alpha-fold co-folding models. So how do you prove this? Because obviously we can't do crystallography. So what we use is a technology called limited proteolysis mass spectrometry. It's a type of omics technology which is trying to uncover the parts of the surfaces of the proteins that are shielded from reagents in the experiment. And this is the type of data you get. I won't go through the details, but essentially what we can do is we can take these peptide sequences and map them onto the structure, onto the surface of the proteins. And this is what we got. So on the left-hand side is the tech-predicted ternary complex structure. And in dark blue on the right-hand side are the peptides that were shielded. So what we see is we can unambiguously prove that this is how the proteins are interacting with one another. We've subsequently taken the structure here and we've used this to optimize the molecules, which AH are synthesized, and I'll show some data on that. Before I do so, I just wanted to show you an example of where some other state-of-the-art models compare to us. So this is Boltz-2, it's a co-folding model. We ran Boltz-2 on the E3 ligase and the target, and you can see that actually the complex that it predicted was completely incorrect. There were no surfaces overlaying with the experimental evidence that we were able to generate. So in a single round of optimization, not only were we able to increase the degradation profile, but we were also able to move from a PK profile that terrible from the literature compound to something where we were still able to detect it after 24 hours. So a really, really nice example of where in just a couple of months, a single round of iteration at O2H, we were able to identify some really optimized molecules. And we also have brain penetration. Since the CNS project, we had some really lovely examples of where it was readily distributed into the CNS. As I mentioned at the start, our focus is around activation, but our tech platform can be used for any type of modality. It can degrade. We can create non-degrading inhibitors and activators. We can look at macrocycles. We can look at small molecules. But obviously, we don't want to try and boil the ocean. We're worried a lot about being focused so that we can deliver in an area and then scale through collaboration. So our internal pipeline is focused and it's unique to us. We have 2 activator molecular glue projects. These are highly validated, notoriously undruggable drivers of neuroinflammation. I probably wouldn't need to describe them to you. You probably know them by their gene names. We have over 300 other global pathogenic loss of function mutations that we're currently evaluating in target ID and starting to run hit ID processes. We've already signed 2 collaborations. Our first is with a top 10 big pharma, and this is for a degrader. Set of degraders in oncology. And we also have biotech collaboration for macrocyclic non-degrading molecules in oncology, starting to sort of demonstrate the applicability of our platform. As I mentioned at the start, we are, and it's important, we did this at the start, we are drug discoveries. We're drug discoveries that design and invent novel tech approaches to solve the problems that we find. We are pioneers in AI and drug discovery. I spent 6 years at Benevolent AI, previously 10 at GSK. A lot of our team come from Benevolent AI, and we span everything from machine learning, bioinformatics, through to chemical biology, medicinal chemistry. This is a snapshot of the team from the summer. We're now 11, but we have a track record of being able to deliver candidates for a range of diseases and novel tech products. In terms of our advisors, I'm pleased to say that we have 2 excellent industry veterans as independent board directors. So Ian Taylor is recently stepped down as president of R&D at Arvinas. In the summer, Arvinas, for those of you that don't know, are the company that created the 3rd marketed drug of a proton. So a degraded drug in this space. And he has a long history in the oncology space and Bayer advisor. I imagine I don't need to describe Jackie to many people here, but she's clearly ex-CEO of Benevolent, ex-head of BBSRC, neurology at GSK, one of the most influential biotech and big pharma leaders in the UK, I'd say. And then from a scientific advisory perspective, we have all people that lead in all the different areas that we need. Krista is a medicinal chemist, CSO at Larkspur Biosciences. They've just dosed their first patients with a degrader for an undruggable target. Catherine's a ubiquitin expert, Francis Crick, Fergus is a machine learning researcher at Oxford, and Myrtle is a neurology expert in Dundee. And we're backed by fantastic set of investors that really span that tech bio space. So our most recent round was led by Daphne, which are a Parisian tech-based investor. Future Planet Capital through UKI2S also supported Pace Ventures out there in I&I in China. So that's everything.
TBD expressing that I won't think of them, but maybe in the audience, sorry, maybe there's a couple of questions. There are definitely questions. No, move on. Thank you very much.